Thank you, Master. Let me share screen first before talking to the people. Okay, I think everyone see my screen. This is okay. My screen is show up. Yes or no?
Yes, Professor.
Okay, thank you. So good morning, all participants, the chair, and first of all, I would like to thank the organizing committee to invite me. I'm very nervous and a little bit exciting to give a talk today. Actually, we should see straight by face. I love to see to have a meeting face by face rather than going online like this. But you know, we have no choice with the COVID-19 pandemic. But hopefully, in the near future, we can meet each other and then we can discuss science face by face. So I'm going to give a talk on my topic gene editing tool in the carcinogenic liver fluke Opisthorchis viverrini. Okay, most of the work that I have going to present to you is the work that's already published in the past 10 years that I start to work with functional genomic of the Opisthorchis viverrini. So it's a little bit own work, but we have a new work, but I'm going to present later if I have a chance. Okay, the content today, not only gene editing tool. In the past 10 years, I have been working on the functional genomic tool in the liver fluke Opisthorchis viverrini. So I'm start with the RNA interference. So today I'm going to give a talk on the development of RNA interference for gene function analysis as well. And recently, the gene editing tool using CRISPR-Cas9 is a new technology that was just discovered and was awarded by Nobel Prize recently. We also done the experiment in the using gene edit tool using CRISPR-Cas9 in Opisthorchis viverrini as well. So the major content would be dealing with two techniques that I have been using in my research. Okay, so let's start with the RNA interference. With why I'm interested to working on the functional genomic, because you know now if you work in the molecular if you work in the parasitology, now research in the molecular parasitology already enter the post-genomic era. Most of the cellular genome of the parasite has been published 10 years ago, like in Opisthorchis viverrini, the genome is already complete and several of the parasitic disease already have genome complete. So the post-genomic era, not only the genome, in addition, the transcriptome proteomics, you know, many of the transcriptome and proteomics study has been studied in many of parasites, generates massive amount of the information. So model tool to investigate gene and protein function should be applied. I'm working in northeastern part of Thailand, Khon University, and the major parasitic disease that we are interested in is the liver fluke that cause cancer. I think most of you in this meeting would know about the human liver fluke. There are three major. All of them now is classified as a carcinogen. The Opisthorchis viverrini affect 10 million, infect 10 million people. Is what classified as a group 1 carcinogen as well, and also Opisthorchis felinius and Chronicis sinensis. I'm working on this parasite, Opisthorchis viverrini. Is a liver fluke that's endemic in sub-Mekong region, including northeastern Thailand, Cambodia, north of Vietnam, and Laos. Many of you may have this problem as well. You know, among these, about 600 million people. And I think many of you also know this. I'm not going to take too much time on the life cycle of the OV, but some of people would interesting that the Opisthorchis viverrini, the liver fluke that caused by raw fish. This is, people who got infected have to eat the raw fish, especially the cyprinid fish. This is the popular fish that infect people in northeastern Thailand have it, koi pla and plasom like this. And the adult worm reside in the bile duct cause pathology, including, after they have long infection, they can turn out to the cholangiocarcinoma. That's why this parasite can cause cancer. That's why we interesting to do the research on this, why this parasite cause cancer, and that's why that's cause high pathology. So as I already told you, Opisthorchis viverrini was classified as a group 1 carcinogen. And even there are several evidence in the laboratory epidemiology showing that the liver fluke cause cancer. But the mechanism, the real mechanism still unclear. But up to date, I think some of you know the major proposed mechanism is including 3 factors that liver fluke cause cancer, including mechanical damage, immunopathology, and I'm interested to the fluke protein, which is especially focused on the excretory secretory protein of the parasite, because a lot of evidence from research, we found several key molecules. For example, Opisthorchis glandulin 1, the Opisthorchis extracellular vesicle, the molecule that Professor Wu just give a talk before me. And then we found some that these 2 proteins is involved in the modification of the cell. They promote cell proliferation and induce the immune pathology, including interleukin 6. And also we also identify the Opisthorchis viverrini thioredoxin that is payroll as an anti-apoptosis. So some of this molecule is involved in the carcinogenesis. So we would like to investigate how important or how significant of this kind of molecule is involved in the carcinogenesis. So to investigate the gene function, we need some the molecular technique that model to investigate the function. RNA interference is one of the model techniques that we use to investigate the function of the gene. So we start to work with RNA interference since about 10 years ago. Before I'm going to tell about my work about the RNA interference, I'm going to introduce you a little bit what it is. RNA interference or RNAi is the method for silence the gene. So if you want to knock down or silence the function of the gene to see if that gene is play the function, you can use this technique. And we have successfully developed the RNAi system in the platyhelminth, including the Opisthorchis viverrini, by using double-strand RNA to induce RNAi. And we can use this in both adult and juvenile worm. I will tell you in the detail how we construct or how we develop this kind of experiments. And we use this technique to validate protection for the target of the drug and vaccine development. For example, we developed the vaccine from the protein tetraspanin EV by using the RNAi to check the function of this gene. So RNAi was discovered long time ago. I cannot remember, but it was awarded Nobel Prize in 2006. These 2 guys, Andrew Fly and Keck Mello, developed the RNAi in free-living nematode C. elegans, and he can knock out some gene and then can prove that RNAi can be used in the organism. So this is the principle of RNAi mechanism how it works. RNAi or RNA interferon is a natural mechanism that used for the organism to suppress some gene that they don't want them to express. And this natural mechanism can be applied in the laboratory. So we use this mechanism for suppressing some target gene. So this is the general process that RNAi working. Start with they will have the double strand RNA, and double strand RNA have to be cleaved by dicer protein enzyme, and dicer enzyme will be incorporated with RSSC complex. After that, RISC, which is specific to one mRNA that they want to cleave. So this RISC will digest or cleave the mRNA of the target, become a very small piece, and then protein can be not synthesized. So we use this technique to mimic in the experiment by we introduce double strand into the liver fluke, and then that double strand RNA will process the RNAi mechanism to stop RNA, to digest mRNA and stop protein synthesis. So from our laboratory in the Opisthorchis viverrini, we have identified several of, we have done the transcriptome and proteomic. And then there are several of the interesting or target protein that found from transcriptome that, you know, normally when we focus on the protein that may be associated with the pathology, we will focus on the protein that parasite secreted, like protease protein, and, you know, most abundant, just like cathepsin F, cathepsin B, cathepsin D, the molecule growth factor, exosome, and microvesicle. And also the tegument protein, which is the transmembrane protein, if it's not secreted, but the tegument is the part of the parasite that attacks to the host system. And also tetraspanin is a key molecule in the extracellular vesicle of the Opisthorchis viverrini too. So we have done RNAi experiment in this kind of protein to see if we can knock out the function of this protein, it will be stopped, or what is will be involved in the pathology of the Opisthorchis viverrini or not. Okay. And all these protein we already know in the literature, or we propose that it should be involved in the pathogenesis of the CCA, and probably can be applied if we can approve that function. We can apply this protein as a drug or vaccine target. So to set up the RNA interferon in the Opisthorchis viverrini, we're using double strand RNA system. So there are two major systems for the RNA interferon experiment. You can use the short RNA, or we call it siRNA, or double strand RNA. In the platyhelminth, there are several people that pioneer in the platyhelminth. They use double strand RNA, and it seems like it's work better than the miRNA, the short strand RNA. So we use double strand RNA. We construct the double strand RNA. Normally we have to find the target of the mRNA, and we have to decide about more than 500 base pair of that gene, depend on what gene, and then you incorporate with the T7 gene specific. So to construct double strand RNA, you can buy the kit. We use the megascript, and then we introduce this double strand RNA into the worms. So to induce double strand, to transfer double strand into the RNA, very easy. The system we use electroporator. You have to have this machine. It's a square wave electroporator. So you prepare this double strand RNA, and then you mix with the worm in the cuvette, and then you shock them with the scale wave. You need to optimize this system a little bit, but from our experience, we use 50 microgram of double stand, and then we shock with this 225 volt for 25 minutes. And then after that double stand will go into the cell. Then we would have to check the RNA expression level of that gene of interesting, protein level. Normally we use QRT-PCR for RNA expression level, and we use Western blot and proteomic for protein level. And the most important thing, after you suppress the RNA and protein level, you need to check the phenotype change or not. It depends on the gene, the target, that gene that you investigate. I will give you an example of our research. So the first RNAi experiment we have done in 2011. We start from that, but we published in 2011. We focus on cathepsin B, and rather on, we have done several genes. For example, the acropolin, tetraspanin, glandulin. So go to the result. We use that approach. We construct the double stand RNA introduced by electrophoresis, and then we check RNA expression. The first experiment we have done is very easy one, because at that time we were very young, and we don't know much, and we don't have much material to do the RNA, not so expert. So what we have done it. We introduced the double stand targeting the cathepsin B in the adult worm in vitro. So we introduce that and then check the RNA expression by real-time PCR. And that was amazing, because RNA interferon can decrease the mRNA expression of cathepsin B after day one after electroporation, more than 90%. And when we went to talk with people, most of them would say RNA interferon is the most effective technique to suppress the mRNA level. But at that time, we do not have material enough, and we don't have time enough. We didn't check the protein expression. But with that mRNA expression decreasing, we using that worm to check after we knock down the mRNA level of the cathepsin B by RNAi. What happened after that? So the cathepsin B activity also decreased. So we using the crusomatic worm from the knockout worm and compare with the control, the normal one. You can see that the activity of cathepsin B is less than the control one. So that means RNAi can be used to knock out cathepsin B, to knock down cathepsin B, both in mRNA and protein function. So that is small experiment. We just do this thing. We publish in a very small journal, Experimental Parasitology. And then next, after we confident to do the RNAi, we focus on the another protein we call tetraspanin. Tetraspanin is a transmembrane protein. We found that tetraspanin is play function in the formation of the tegument of the worm. So we hypothesize if we can knock out the tetraspanin protein, can make the tegument of the worm malformation. So tetraspanin can be one of the vaccine target. So if you can produce the antibody against the tetraspanin, that can be killed the worm. So we using RNAi interferon, and then we can prove that RNA interferon can use to knock out the tetraspanin 1, 2, and 3. And then after knockout, we take that worm and then check with the transmission electron microscope. So you can see this is the section of the worm, and this is the normal tegument of the adult worm. You can see after we introduce double stand of the Opisthorchis viverrini tetraspanin into the worm have degenerate malformation of the tegument here. And also happen in tetraspanin 2 and tetraspanin 3. So that means suppression of the tetraspanin by using RNA interferon also works. And we also using the RNA interferon to check if we knock out the tetraspanin 2, which is the major protein in the extracellular vesicle of the OV. So this is the hepatocyte cell line H69 normal hepatocyte of the human. So the blue color is the nucleus of the cell. So in the normal control, that means the normal worm, we using the extracellular vesicle incubate with the hepatocyte cell. You can see the clean color. That means the EV in endocytosis or internalize into the cell. So, and this internalization can cause pathology. We know that this internalization can cause pathology in the cell, increasing the interleukin 6. Then we block the worm. We using the RNAi to block the tetraspanin. You can see that after block tetraspanin, the endocytosis of the EV is less than in the control. So that means the tetraspanin, we can prove the function of the tetraspanin that play a critical role in the endocytosis of the extracellular vesicle by using the RNAi interference. And also, if you see in the messenger RNA level, they also decrease. And this is another example. This molecule called acropolin. So we have shown that after introduce double-strand RNA into the worms, the adult worm, the messenger RNA expression is less than the control. And we try to check if mRNA of the acropolin was effect after the knockout. So the experiment was very easy because the acropolin is the water channel control. That control the water get into the worm. So normally worms have to stay in the isotonic solution. If you incubate the worm in the water, that is hypertonic solution, the worm will get swelling. So we hypothesize if we block the acropolin, which is control the water into the worm will not swelling, and the result show up as we expect. The worm that's knocked out with the acropolin double-strand RNA, they're not swelling compared to the control. So this experiment is an easy, cheap one that we can prove that acropolin is a water control channel after we knock with the double-strand RNA. So, and then that is the early phase that we working with the RNAi. We still cannot find a very nice molecule that can be used the RNAi for investigation the pathogenesis of the Opisthorchis viverrini. In about 2008, we identified the growth factor protein called Glandulin 1 from the proteomic projects. And then we found that Glandulin 1 is play a role in cell proliferation, which is one of the major part of pathology of the OV-induced cancer. So we hypothesize if we can block the Opisthorchis Glandulin 1 by using RNAi, we will reduce cell proliferation. And that time, we only can do in vitro. That means in the test tube only. We cannot knock out the worm and then put into the hamster or put into in vivo. So what we have done in the first experiment, we try to prove if Glandulin 1 is involved in cell proliferation by knockdown using the double-strand RNA. And the result is the same. RNAi is always work well. using the same system, using double-strand RNA, 40 to 50 microgram electroporate into the worm, and then culture, check the mRNA. It's almost 100% decreasing. If you see from the graph, this. So OV glandulin 1 start from day 2, the messenger RNA it start to decrease. Then we take that worm after knockout and then co-culture in the cell line, both the normal normal cholangiocyte cell and cancer cell. The worm that were knocked down by double-strand RNA, OV glandulin 1, have less cell proliferation significantly in the middle. So this is the baseline. This is the normal worm we call mock control. Mock control is mean you electroporate without double strand. This is negative control without anything. So we can prove that knockdown of the glandulin 1 can inhibit growth of cells proliferation. At that time, we use the WST assay. And we also using the wound healing assay to check if knockout of the double-strand glandulin will show the less wound healing, because in the normal cell, the glandulin is the growth factor. It will stimulate the cell to heal the function easily. So in the negative control, they're not healing. And then in the double-strand, they're healing slowly than the mock control. So that also proved the function of the OV glandulin in the wound healing process. So in summary of the RNA interference for the OV, that we have been done in several target genes. I would encourage you to, if you want to investigate the gene function, electroporation of the double-strand RNA can induce RNA interference to knock down the target gene in the Opisthorchis viverrini. And I think it's also work in the another helminth as well. But the level of suppression of mRNA protein is vary, depend on the species and depend on the gene. If you remember the result of my experiment, some of the gene like tetraspanin, only about 60% or 50% can be reduced. But some of the gene like glandulin 1 have a very high efficiency. But anyway, the RNAi in Opisthorchis viverrini can be induced by using double-strand electroporation. But the problem of the RNAi is early listable effect. After you introduce the double-strand RNA into the parasite have defense mechanism, and they think the parasite try to get rid of double-strand out because they want to survive as a national mechanism of them. So the knockdown effect by RNA interference is transient, short duration only. So if you want to do the functional analysis just for the short time or in vitro in the test tube, you can use the RNAi for your research. But with our research, we focus on some molecule. For example, we focus on the glandulin 1, which is we would like to investigate the pathology of the pathogenesis of the Opisthorchis based on that molecule. So we would like to show if that molecule was only effect in vivo or not. If we use the RNAi and then we introduce the RNAi into the worm and infect to the host, the RNAi will disappear. So the worm will not, the knockout of the worm will not persistent. So we have to think about the new technology for suppression the gene. And luckily, in about last 10 years, the CRISPR-Cas9 was discovered, and there was won the Nobel Prize in 2020. And the CRISPR-Cas9 is the method for genome editing. So CRISPR-Cas9 can edit the genome and then can make the mutation to the gene of the interest. So we interesting to apply this technique to investigate the function of the molecule, especially we focus on the Opisthorchis viverrini Glandulin 1. So the CRISPR-Cas9 is a genome editing tool. So generally, CRISPR-Cas9 is a natural mechanism of the bacteria. I tell you later. But the CRISPR-Cas9 is a genome editing tool using Cas9. Cas9 is an endonuclease, and Cas9 is generate double-strand break to the interesting of the genome. And then Cas9 will bind to the guide RNA. Guide RNA is the area or the target that Cas9 cut and then make it mutate. So normally guide RNA is contain only 20 nucleotide, and that nucleotide will flank with the sequence called PAM, which is characteristic of PAM is NGG. So any sequence that start with N and any of the nucleotide, I'm show you later. And after that, once the guide RNA, the Cas9 go to this guide RNA, double strand break will repair, and then we'll make a mutation to make easy understanding. I show you later. And after that, the Cas9 will delete or insert and mutate the DNA of the target gene that you're interested in. So like I told you before, then the Cas9, CRISPR-Cas9 is natural system. Is natural system of the bacteria to protect themselves from the virus. So in natural, when the virus go to infect the bacteria, go to the chromosome, the bacteria will produce the guide RNA to recognize the DNA of the virus. And then this guide RNA will be incorporated with Cas9. Cas9 is endonuclease. So the guide RNA will target the virus and then cut the DNA of the virus. So the virus cannot infect. So this is the normal mechanism. And the scientists found this normal mechanism can be used like an engineer CRISPR-Cas9 we can produce guide RNA and then target to any gene that you want to mutate them. So what we are doing is the same system of this. We need to construct the plasmid that express CRISPR-Cas9 and insert the target of your interesting gene. So in this case, we start with the OV Glandulin 1. So we have to design the guide RNA based on the Glandulin 1 and then construct this plasmid and introduce into the parasite. And then this guide RNA will work under this CRISPR-Cas9 system to mutate the gene of interest. So this is the idea. So this is the target. In this case, we using the OV Glandulin 1. So you need to identify the target gene. So the first step that you can decide the CRISPR-Cas9, you need to have the genome sequence or genomic sequence of that gene of interest. So in this case, the genome of the Opisthorchis viverrini already sequenced. So we contact the person who sequenced that, who is working in the Melbourne University. So and ask them, can you provide me the sequence of the OV Glandulin 1? And then we decide the target gene. So the target gene, like I told you, is only 20 nucleotides, but have to flank by PLAM. And then this sequence flanked by PLAM. And then we introduce into the worm, Cas9 will work by with the guide RNA. Guide RNA will cut both strands of the target and then make mutation. So it will repair without the scar, but this mutation will occur after the guide RNA was cut. So there will be deletion mismatch, but the gene will be mutation. So after the gene mutation, the function of the gene will change. We expect the, the protein will, the mRNA cannot express in the normal manner. So in our research work, we focus OV granulin 1. So this is the Opisthorchis viverrini. And then we hypothesize at this moment, current study, we know that OV granulin 1 induce cell proliferation. So we hypothesis if we knock out the glandulin 1 and then put the worm into the hamster, there will be less pathology when the glandulin 1 was knocked out. And then if we can prove that the glandulin 1 will be one of the vaccine target or drug target to prevent cancer. So this is the hypothesis of us. Then to construct the OV glandulin 1, we need to so we got the sequence from Neil Young in the University of Melbourne. So we found that glandulin 1 have 3 axons, 1, 2, 3, and then 2 axons. The size is around this. So we focus on axon number 1. So we decide guide RNA, which is only 20 nucleotide. And this is the sequence of the PAM. This is the key designing procedure of the guide RNA. You need to have target site fang with PAM. PAM is NGG. That means any nucleotide and GG. So we expect that after they cut by CRISPR-Cas, they can have insertion, deletion, or mismatch on this target site, and then mRNA cannot produce. So what we done is we clone this guide RNA into the pCas9. So you have to buy this kit in the in vitrogen company. And then after you construct this, you will have guide RNA combined with the Cas9, and then you introduce into the parasite. And then the DNA will go into the nucleus and then transcribe and then cut. The guide RNA will work and then we'll cut the target site. This will happen after you introduce the guide RNA in the guide RNA, the CRISPR-Cas9 into the cell. So we're using square wave electroporation, same as we're using in the RNAi. So what happened after that? We start with adult worm first in vitro to check if CRISPR-Cas9 can knock out glandulin 1. So we culture the worm. So we electroporate the worm with the double stand, and then we check the mRNA. So the results show that the mRNA of the worm that electroporate with OV glandulin plasmid have less. You can see this is the pink of the OV glandulin 1 fluke. That means this one was electroporate with the double stand with the CRISPR-Cas9 plasmid. So this show the mRNA level. After 4 days, the mRNA level almost totally suppressed. So that means we are successful to knock out the mRNA level using CRISPR-Cas9 transfection. And we also check protein by using antibody against this using the Western blot technique. And the results are the same. Not only mRNA was decreased, the protein expression also decreased. So it's almost 100% in the plasmid transfect group. So that means the CRISPR-Cas plasmid transfection is really work well. Then we try to investigate after knockout in vitro in the culture. So we transfect that worm with the plasmid again, and then we culture in the normal H69 or hepatocyte cell. You can see the cell proliferation is less in the knockout OV glandulin 1. So almost 100, almost 100% in the OV glandulin group was less cell proliferation compared to the control. The green and the blue one is the control. This is the baseline, nothing at all, and this is the suppressed worm. And also wound healing. We can prove that the OV glandulin 1 knockout by CRISPR-Cas9 have less wound healing. So after that, one we can prove CRISPR-Cas9 is work well in adult worm, but we still cannot prove if this worm will be effect in the host or in the real pathology or not. So we need to do something. So we cannot knock out the adult worm and then implant to the hamster. So we think it would be okay if we can knock out the metacecalia, which is the juvenile worm, and then we infect, and then if the suppression or the mutation still persist until they are adult, these glandulin still not expressed as well. So we start with using metacecalia with NCIS, and then it's not working because the double stand cannot introduce into the cyst wall. So we have to change the strategy. We exist this metacecalia and then electroporate with the plasmid CRISPR-Cas9 and then infect to the hamster. So first of all, we need to check if it's work in the metacecalia. So in the exist metacecalia, the expression level was decreased almost 100% too. So we can prove that we can use the newly exist juvenile or newly exist metacecalia, transfect with the CRISPR-Cas9 and then infect into the hamster. So we make 3 groups. So non-infect, infect with the normal wild type, and infect with the CRISPR-Cas9. And we infect them to dilation time to see short-term pathology and long-term pathology. For 14 days, we will check the inflammation. So after we infect with the 100 OVNEJ 14 days, we get the liver and then stain with the H&E for the cell proliferation pathology histopath, and then we stain with picocilious and anti-actor 2 for the fibrosis. And for 60 days, we also doing the same thing. And at the 60 days, we also check, we separate 2 hamsters to see if the worm still survive after they were transfected with the CRISPR-Cas9 or after they were knocked out with the OV glandulin 1. So the hypothesis is should be less pathology in the knockout or in the group 3. So in the group 3 that transfect with the CRISPR-Cas9, H&E and picocilious should have less pathology and the worm still survive. And this is the result. When first time I saw this result, I was so happy at that time because this is real. So this is the normal. This is the normal hamster. So you can see the epithelium bile duct, just 1 layer. And then if the hamster were infected with the OV for 14 days, the epithelium bile duct is very thick. So this is the picture of the worms that glandulin knock out. Almost the epithelium bile duct is thinner, look like a normal, almost normal. So we have a hope. So we do further investigation. So the thickening of the bile duct is less in the knockout. So that means glandulin is really play a role in cell proliferation induction. So about 11 times reduction. And then at 60 days, people who working with pathology of OV would know 60 days, the cell proliferation would turn to the fibrosis. And first of all, we check if the worm is still survive. And then we can see in the group of the hamster that infect with the transfected worm, the worm is still have almost the same number after infect. And then we check the mRNA level of that worm. So they have decreasing of the expression level in the pink color. This is the knockout group. So that means the worm that still survive in the hamster have less expression level of the glandulin, about 81%. And we check the mutation of the gene by using the next-gen sequencing. We take that worm and send it to the sequence to see the target. There's about 2.7 mutation. Some of them low, some of them medium, and some of them high. Not similar, same mutation level. And then the last result, this is an uninfected worm. So you can see the epithelium bile. There is no fibrosis at all. This is the worm with the white type. There are thick, this red color is a thick of the fibrosis. And in the knockout, very less fibrosis. So we can prove that the glandulin 1 knockout by CRISPR-Cas9 show less pathology, both for the cell proliferation and fibrosis formation, which is one step of the carcinogenesis. And this is the same thing for investigate the fibrosis, but use another staining using the anti-actor2 or SMA to see the fibrosis, which is less in glandulin 1. So you can see this is a high fibrosis in the wild type, and this is the knockout glandulin 1. So it's about 94% less. So from this, using CRISPR-Cas9, we found that liver disease was remarkable decreased after we infect with gene-edited liver fluke. So less pathology when we knock out the glandulin 1, including epithelium dysplasia in 14 days and reduced fibrosis in 60 days. So that means the worms that were mutated, the glandulin 1, have less pathology. So this is the picture. This. So we publish in eLife. So in conclusion, CRISPR-Cas9 has been applied to several parasites. So, you know, now you can use CRISPR-Cas9 to study in several aspects of the parasite, and it's not an complicated experiment. You can do it easily. So in the future, the system we're using now is just CRISPR to mutate the target gene. But to apply the CRISPR to become more advanced, we now develop the CRISPR knock-in system. That means not just only mutate the target gene by random. We can specify what kind of gene we need to do. So there are the recent publication in Nature using CRISPR-Cas9 to do the gene drive to control the parasitic disease. So this group is modifying the mosquito that is a vector of the malaria, and then try to spread that in the environment. So this is the new trend for the using CRISPR-Cas9 in the future. Okay, I think I used the time properly, not too much. And I would like to, this work is a collaboration in between 3 countries, Australia and USA. This is Alex Lucas from James Cook University, and this is Michael Small, who is originally working on the glandulin. This is Suchitra, who working on the extracellular vesicle. And this is the student who working on the CRISPR-Cas9, Wannapon and Paul Blinley, Victoria Mann, his wife, and Meredith, who is the person who draw that cartoon of hamster. So thank you very much for your high attentions. I stop share. So welcome for any question. I pretty use a lot of time because too many things to talk, but still welcome to the question. Maybe I have 1 or 2 questions. That's enough.
Thank you very much, Professor Thewarach, for a very informative talk. There was one question in the chat box from Dr. Opisthorchis. As you mentioned, theta-spanine protein is the surface of the worm. Do you have plan to do CRISPR-Cas9 with the theta-spanine gene?
We already did it in the theta-spanine, and then the manuscript is under review in the JEV. So we found the same thing. As if you remember, theta-spanine is one function is formation of the tegument. We show that knockout of the theta-spanine using RNAi, making the worm tegument deformative. We found the same thing using CRISPR-Cas9. And also, we also found the same thing after knockout theta-spanine CD63 group in the Opisthorchis viverrini. They have less EV secretion and EV internalization into the cell. So CRISPR-Cas9 also work with the theta-spanine, same as the RNA interference.
Thank you, Professor. There is also another question in the chat box from Kaen.
Why do we have the dicer system? I think most of the organisms have RNAi system. Dicer was shown in the proteomics data too. So, but dicer is an RNAi pathway. I think most of the organisms have RNAi interference, including miRNA and double-strand system.
Okay. Thank you. Now we are running behind the schedule, but this is the last question. I saw Professor Wu raise his hand.
Okay, sorry, I have a question. And whether it's possible to confirm your knockdown in situ and in vivo, use the immunostaining with the anti-glandulin 1 antibody. Is that possible? Such as immunostaining your section, and you use the antibody and see the knockdown is really in situ and in vivo.
That is a good question. We cannot do in situ, but we can take that worm and then do immunolocalization if there are no glandulin. But we haven't done it in the glandulin 1. We have done it in the TSP2-1. So we can stain, but we not really stain for the theta-spanine 1. We stain for the EV. So we're using transmission electron microscope and then see the particle of the EV. So we are doing at this moment, but we have the problem of the quality of the EM. So the reviewer not believe that is the real EV.
Okay, another question, sorry. In the future, whether it is possible, this kind of knockout can carry down to next generation, such as agar. That agar is knocked down. Then use that agar to set up the knockdown clone of the parasite. In the future, it's possible or not?
I think if we can find support or gland, we can do in the future. Because this kind of work is you have to have like a big collaboration and big plan, and we have a good collaboration who are also expert using CRISPR-Cas9 in another lab. I think we will do more advanced.
Okay, thank you.